Okay, so welcome back to this next video in which we are discussing cancer development. So we are currently in the process of discussing the changes which need to accumulate in these cells, in this tumor, that are going to progress us towards becoming cancerous cells. Okay, right. Now, I want to stress that the tumour that we've got at the moment is still a benign tumour. It's not malignant yet. None of the cells in this tumour yet are capable of invading the surrounding tissue or metastasizing to far-off locations. So at the moment, it's still a benign tumour. Okay, and really what we're now doing with this expanded view of the development of cancer is we're filling in the gap between that initial mutation which caused the cell to over-divide and then those final changes which then cause it to become over-motile, basically. And I'm saying mutation, but of course I should really be saying heritable change, because it can also be epigenetic changes in the structure of chromatin, which are heritable, which are causing the change in protein function, which is then driving the oncogenic phenotype. Okay, right. We've seen that the first mutation was that the cells led the cells to over-divide. Then the second mutation was that you got some sort of genetic instability, which then meant that mutations in the genes would occur more frequently, and that means that you're more likely to accumulate the necessary changes which are going to progress you towards cancer. So that's generally one of the early mutations that you get in this process. And we're now discussing other changes that are going to occur towards as we progress towards becoming fully cancerous. So one that we've discussed is this Warburg effect, where the cells are going to acquire changes in their metabolism that better suit them to surviving in the hostile environments of a tumour. Okay, right. The next one I want to discuss is that we're going to need some changes in proteins that are involved in the cell response to stress. Okay, so I now just want to talk about what the cell's response to stress is. And before we can discuss what the cell response to stress is, we need to firstly define what cell stress is. Okay, so basically, the cell response to stress is a response to the cell having very peculiar properties, and the peculiar properties that count are that it's over-dividing. Okay, so cells that are dividing very quickly, that is not a good thing generally. So that is something that sounds alarm bells within the cell. Okay, so this is one of the characteristics of cell stress. In addition, DNA damage, okay, is counts as cell stress. Okay, and finally another thing is bizarre metabolism. Okay, things that we have seen, basically, we have got these cells which are over-dividing, we've got cells which have DNA damage because they've got genetic instability, and then we've got these bizarre metabolisms. We're going to have, lactate building up within the cells and things like that. Okay, all of these things, they are odd behavior for cells. Now, cells have special pathways within them, okay, which are designed to become activated in response to cell stress. Okay, so cell stress is really a cell showing bizarre behavior. Okay, and the response to cell stress is that you stop the cell from dividing, and if necessary, you cause that cell to commit suicide. So basically, this is a mechanism that all human cells have, or at least all normal human cells have, for protecting the multicellular organism. Because if a cell starts displaying bizarre behavior, i.e. if a cell becomes stressed, the idea is that it will activate this pathway, and we're not going to go into the details of the pathway, but we just need to know that there is this pathway. Okay, it will activate this pathway, and what this pathway will do is it will stop the cell from dividing. Okay, stops division. Okay, so it stops it passing its bizarre behavior on to progeny. Okay, the last thing you want is this cell that's behaving ridiculously strangely, making more cells that are behaving ridiculously strangely, which is exactly what's happening in this tumour at the moment. Okay, so this really is a pathway to protect you from cancer, okay, that all cells have. Okay, and it also, if necessary, will make the cell commit suicide. And the fancy word for the pathway of programmed cell suicide is apoptosis. So this pathway is a pathway for protecting the multicellular organism. If you get cells which are behaving in bizarre ways, they have this inbuilt pathway that tells them to die, or at least to stop dividing. Okay, so this is to protect the multicellular organism. This is a horrible pathway as far as our cancer cells are concerned, okay, because they are doing the behaviours that will activate this pathway, okay. So this pathway will be taking its toll on these cells, okay, and some of them will be apoptosing because of it, okay. So at the moment, this pathway is limiting how big this tumour can get. It's doing its best, it's killing off some of these cells. I mean, it's not perfect, it's not going to work perfectly in every single cell, which is why we've got some that have managed to get around this, okay, but it will be limiting the growth of this tumour. So generally, in fact nearly always, what happens is that you get problems with this pathway. Specifically, there's a very famous protein that's involved in this pathway. In fact, it's pretty much the first protein in this pathway, known as p53. This becomes active upon cell stress, and then it activates these two things. It activates the prevention of division, and it activates apoptosis if necessary. And p53 is given a very grandiose title. Just as Mick was called the cell's most powerful mitogen, p53 is known as the guardian of the genome. Okay, so it's a very, very powerful protein, and it's found mutated in around 50% of cancers, okay, and that's only one component of this pathway. There are other components of this pathway, so this pathway is found mutated in nearly every single cancer, okay, and it has to be, because it really is the pathway that stops cancer. It's your safety mechanism. So to get cancer, what has to happen is at some point they have to stop this pathway. They have to get changes, heritable changes that stop this pathway from functioning. Okay, so let's now say, and again I want to stress that I'm giving this an order. I'm saying this one occurs first, this one followed by this one, followed by this one. I'm doing that just to help you understand. In reality, these different mutations can occur in different orders, okay? But I find that the best way to explain this is to give it an order, because then at least people have something to visualize, and then tell them afterwards that in fact you can mix up the order. Okay, right, so let's now say that one of these cells with the altered metabolism here, with the genetic instability, and with the overproliferation here, this one that I'm outlining in black, is now going to get some heritable change, which will most likely be a mutation that's going to completely stop this pathway. Okay, now that cell is going to do so much better than all the other cells, because all the other cells had this pathway on, okay, and this pathway was fighting them. It was fighting them really strongly, okay, because they were showing this bizarre behavior. They were cells that were stressed, okay, and this pathway was trying to stop them dividing, and it was trying to make them die, okay, but they were still fighting their way through. They'd survived, and they were dividing a little bit, but now we've got this cell that no longer has that problem, okay, so it's going to be dividing much more rapidly, okay. It's suddenly going to have this break taken off it, basically. It's no longer going to be fighting this, and it's going to be doing much better than the other ones. Okay, so we're going to have another clone of cells then arriving. So let's show this. So here is our tumour, and our tumour will be getting bigger and bigger, by the way. Okay, so our tumour is growing, and the other thing that I should stress is that this is happening over years. The successive acquisition of mutations is happening over years. In between the arrival of these new mutations, you will have the arrival of mutations that are totally useless. You'll have, you know, as I said earlier, mutations in complex 4 that just kill the cell, okay, mutations in everything pretty much, okay, that just have all sorts of strange and weird effects, but it's these specific mutations that are going to propagate you forward in the progression towards cancer. or in the progression towards producing cancer cells. Okay, so let's show cells here. Okay, and I'll just divide it up like this, because I don't really want to show any more of them split up as orange. Well, I do want to still show them as colours, but I want to get across the fact that some of these orange cells are going to start dying, because they'll be outcompeted by some of the later cells. So, let's have the newest strain of cells in not yellow. Yellow won't show up. So this new strain of cells, which are the ones which have suddenly lost the cell stress response pathway. This is a fantastic new clone of cells. This is going to do fantastically for itself, and it's going to make itself at home and take over pretty much, because it's out-competing its neighbours. Okay, so Darwinian evolution all the time. Okay, so here they are, they're doing very well. Okay, and they've pushed aside the previous ones. So here are the turquoise ones now, which are struggling to survive. So these are the ones which, remember, had the Warburg effect. They had the overproliferation, they had the genetic instability, but they don't have the lack of this pathway here, and that was a bad idea, because turquoise and blue aren't really showing up as particularly different, especially in this light, but never mind. Okay, then we've got a few of these purple ones still clinging on for dear life, okay, but they're gradually being outcompeted by the blue ones, and then we've got the orange ones here. Okay, which are the original ones, and you'll still have a few of those. Okay, so this again is that concept of intra-tumor heterogeneity. In this benign tumor, we have many different types of cell here, okay, that are on at different levels towards being cancer cells. Okay, so we've now got our blue clone of cells, which are these cells which have mutations in this cell stress response pathway. They have a mutation, or at least changes, I should say, heritable changes in the metabolism. They have the Warburg effect, they have the genetic instability, and they have the overproliferation. Okay, right, now the final thing that I want to mention, the next change that I want to mention, is something known as replicative senescence. Okay, so this is the portion about the telomeres. Okay, right. So, replicative senescence then. Okay, so, basically this is something that applies for that second type of terminally differentiated cell that I talked about at the beginning. Okay, so remember I told you that there were 3 types of terminally differentiated cells. There were the ones that were totally incapable of dividing. You could not renew if you lost them. The heart and the brain. Okay, then there were the tissues that were capable of dividing. Okay, so remember there were the smooth muscle cells, the fibroblasts, the epithelial cells of certain tissues such as the liver, the pancreas, the not the gastrointestinal tract, the kidney, the breast, the prostate. All of these sort of epithelial tissues, they were capable of dividing and renewing themselves. And then there was that third type of terminally differentiated cell, which were the epidermis, the intestinal epithelium, and the blood cells, which relied on stem cells to divide and produce new versions of those cells. Okay, for the second type of cells which can divide, okay, in the adult human, there is this property known as replicative senescence. Okay, so it's also called, by the way, the hayflick limit. Okay, so let me just explain this concept to you. So basically we'll illustrate it with the example of a fibroblast. So if I take a fibroblast here, okay, remember a fibroblast is a cell that you find within connective tissue. It secretes the components of connective tissue, so it secretes things like collagen. Okay, so we'll have our fibroblast here, and I'll colour in this fibroblast in turquoise here. Okay, so basically you take a fibroblast here, and you try and culture it. So let's put it in a culture flask with loads of nutrients, loads of space, a nice sort of floor to attach onto, okay, and you're letting it divide and divide and divide, and you need to give it growth signals as well. So you put the growth signals in, and it starts to divide. Okay, so it will firstly divide into 2 cells. Okay, so let's just draw them a little bit smaller so that I've actually got space. Okay, then these ones will divide in 2. Okay, so each of these progeny will divide in 2, And as you go along, the number will double and double and double and double. Okay, the question is, can this process go on forever? Well, of course, the answer is no. Okay, firstly, the blunt answer would be, well, surely the flask is going to run out of space eventually, and therefore it can't just go on and on. But then I can say, okay, what I will do is I will remove all the cells from the flask. I'll harvest them from the flask. I can detach them from the flask using trypsin generally. Okay, and then I'll separate them out and put them on new flasks. Okay, can I then make more flasks of cells? It's a very key experimental question. Can I just continue propagating my cells on and on and on and just make more and more cell cultures? Okay, well, the answer is no, basically. The fibroblasts will stop at some point. So imagine they've got a massive great flask, which, they could never actually full. So we don't have the problem of running out of space. They would stop anyway at some point. Okay, so each time you double, okay, the progeny are getting slightly closer to this point where they will stop. Okay, and generally for fibroblasts, it's somewhere in between 30 and 50 times that they will divide, okay, or that the population will double, and then they'll stop. So basically, this fibroblast, it can undergo this division process 30 to 50 times. Now, how many cells will you have by the end of that? Well, each time you divide, you are doubling the number of cells. Okay, so by the end of this, you will have, let's say it divides 30 times, you'll have 2 to the power of 30 cells. And when you finally have that 2 to the power of 30 cells, then none of those cells now will be capable of dividing. It will stop. Okay, so that is what is meant by replicative senescence. Eventually, after an original cell that you took has divided a certain number of times, it will stop, and all of its progeny that it's produced, all 2 to the power of 30 of them, none of them will be able to divide anymore. Okay, that is what is known as the hayflick limit, or replicative senescence. Okay, the cells are said to have gone into replicative senescence because they are no longer replicating. Okay, it's the hayflick limit because it was discovered by hayflick. Okay, and it's the limit to how many times a cell population can double, basically. Okay, right, so what is the origin of replicative senescence? Well, basically, every time a cell divides, it has to copy its chromosomes. Okay, so if I show one chromosome here to illustrate all 46, but this is happening for all 46. Okay, so here is one chromosome. Basically, at the ends of your chromosomes, you have special regions known as telomeres. Okay, and I'll show these in blue. So these regions here are the telomeres. Okay, I'm going to make this a little bit more obvious. I'm going to make them out of proportion to the chromosome, basically. Okay, so this strand here, this is meant to represent a piece of double-stranded DNA. So remember, fundamentally, a chromosome is just a huge, great piece of DNA. Okay, so this line is representing the double-stranded DNA. I could, if you like, draw 2 lines to represent the fact that it's a double helix, but that's just more effort, basically. And because I'm going to have to draw this many times, I'm not going to do that, so I'll just draw 1 line. Okay, now I'm also going to make the telomeres originally much longer than they actually would be. So they're out of scale, basically, to the size of the chromosome here. So this is the normal portion of the chromosome where all of the genes are, and then on the ends you have these much shorter portions, but shown massively here, okay, which are the telomeres, and they should be about the same length on either side. Okay, right, so this is the case for this original fibroblast right at the beginning. Okay, now every time this fibroblast divides, the telomeres will get shorter. Okay, so this fibroblast is going to divide, and the 2 daughter cells here, they will both have slightly shorter telomeres. Okay, so a little bit will get chopped off. So let's say this final bit at the end will get chopped off when you divide. So the purple bit will remain unchanged, but now the telomeres, okay, here and here, are slightly shorter than they were originally, and that's the case in both of the daughter cells. Okay, so both of the daughter cells now have shorter telomeres. Now, when each of these daughter cells divides in 2, the same thing will happen. Okay, a little bit will be chopped off the end, and both of the daughter cells of these two daughter cells here, all four of them here, will now have slightly shorter telomeres. Okay, so here we go, like so. And then this is the middle bit of the chromosome, and here are the telomeres here. This will continue on and on as you double the population more and more. So the further you go along this great expansion here, the shorter the telomeres of the cells in that population now are going to be. Okay, until right at the end, once you've got to the final massive great population of 2 to the 30 cells, you will have no telomeres left. Okay, you will just have the main bit of the chromosome here in red. Now, if you continued on dividing, you'd start chopping bits off the actual chromosome, which is not good at all, okay? That is not what cells want to do. So as soon as they reach this phase, what happens is they go into replicative senescence and they stop dividing, okay? Right. So that's the origin of the hayflick limit, the telomeres getting shorter and shorter and shorter. And in all of these terminally differentiated cells of the second type, which are capable of dividing, you have the hayflick limit, basically. And this is a pain if you're trying to grow these cells, if you're trying to culture them. Okay? So, not all cells have this in the adult human. The stem cells are an important exception. The epidermal stem cells and the intestinal stem cells and the hematopoietic stem cell, which we'll come across more later, okay, these don't have this problem. Okay, now why do they not have this problem? Well, it's true that every time they divide, their telomeres do get shorter, okay, but they have an enzyme which is capable of extending the telomeres, okay. They have an enzyme called telomerase, which is basically capable of adding bits back onto the telomeres and effectively just continues to re-lengthen the telomeres every time they shorten, basically, so that stem cells effectively can divide as many times as they want. They do not have a hayflick limit. Okay, right. Cancer cells also get around the hayflick limit. They remove it. Okay, and the common way that they do it is through the activation of the enzyme telomerase. There are other mechanisms as well. They need some sort of inheritable change that means that they get around this problem of their telomeres shortening every time they divide, okay, so that they then never reach replicative senescence and they are effectively capable of dividing indefinitely. Okay, so that's the final major thing that I want to outline as a change in the phenotype of the cells that you need to have acquired on the route to getting ready to finally become invasive. Okay, so going back to our model here, here's our tumour at the moment. In one of these blue cells now, okay, you are now going to get some heritable change, maybe an upregulation of the amount of telomerase, okay, which is now going to mean that this cell is capable of extending its telomeres and makes itself never reach replicative senescence. It won't have a hayflick limit. Okay, that too is going to help this cell divide more and more, because you know all these cells stemmed from one cell. Okay, so we must be getting close to the hayflick limit now. We've been dividing and dividing and dividing and dividing. We will be getting close to the hayflick limit. The telomeres will be shortening. Okay, so we now need one of these cells to get a mutation or some potentially epigenetic change that then is going to lead to us being able to extend the telomeres back up. So let's say it's this cell here. Okay, now that cell is going to continue on dividing when the others are going to start having to stop dividing because they've reached replicative senescence. Okay, so this one's going to get around the hayflick limit, basically. Okay, and it's therefore going to take over the tumour as the others all stop dividing, basically. So let's finally colour that one in what colour should we show it? Well, I'll colour it in light green. So let's now show our tumour that we've got so far. Okay, so here it is. So now in this tumour we're going to have a lot of these new light green cells, which are the ones which have activated telomerase, or through some other mechanism have got round this problem of the telomeres shortening and therefore can divide indefinitely. Okay, so let's colour these in light green. So we'll have these ones here, and we'll have a huge great cluster of them basically. Okay, now I will stress once again that we are not yet malignant. We are still in the benign stage. This is a mass, okay? It is a tumour, but it's not invasive yet. We have not undergone the mutations or the epigenetic changes which are going to lead to us becoming invasive. Okay, right, so here are the new cells which have all of the changes that we've accumulated so far. Then let's put some of the blue cells in, which have all of the changes apart from the removal of the hayflick limit that we've just discussed. Okay, so here they are. Then prior to the blue cells we have the turquoise cells, okay, which are the ones with the Warburg effect, so the Warburg change to the metabolism, but not the change to the the cell stress pathway. Okay, then we have the purple ones which have genetic instability, okay, and also of course all the previous mutations to that, which are the overgrowth. And then finally we have the orange ones which just have the growing too fast. Okay, right, so here is our tumour now, and now that's the end of the mutations or epigenetic changes that I want to discuss that are in the progression towards the point where we're now going to undergo the change that then leads to us becoming invasive. So this is still a benign tumour. Now, in this mass of green cells that we've got, in this clone of green cells, what's going to happen is in one of those, you're then going to get the mutation or the epigenetic change that then leads to you becoming invasive. You're then going to make a new population of cells which are going to invade inwards. Okay, so let's show this now. So we'll show this in red here. So we've got these new cells in red here, okay, which now have acquired the invasive ness, basically. So remember, they might have lost some sort of cell adhesion molecule, okay, so that they no longer have the cell-cell interactions or the cell matrix interactions that hold them in place. And now they are going to break through the basement membrane, okay, and they're going to move into the surrounding tissue. Okay, and they're going to start destroying the surrounding tissue. They're going to break into blood vessels and start chucking cells into blood vessels, and then they're going to set up secondary tumours far, far away. Okay, and it's those secondary tumours known as metastases which are often fatal. Okay, so that is the more in-depth development of cancer then. We've discussed a lot of the changes that you need to have acquired before you can become full-blown cancer cells. Okay, so cancer cells, when you look at them, they generally have all of these properties that I have now described to you, and it's therefore believed that long before they gain the capacity to become invasive, they get many of the other properties prior in the developing benign tumour. Okay, so it's this gradual accumulation of these heritable changes which progresses you towards this cancerous phenotype. Okay, and only once you have all of these properties can you truly be malignant. Only once you've got all of these properties can you truly survive in harsh conditions and invade and divide out of control and cause the damage that cancer cells cause. Okay, so what I do want to stress is that I have put them in a specific order, okay? This is not necessarily the specific order that they occur in. Those changes, those mutations can occur in different orders, basically. Okay, I've put it in that order to make it make sense, and so that I have something that I can at least describe, but in reality you can change the order. Okay, and there are other things that will change within these cells as well that I haven't outlined, but these are the main changes that are occurring in that progression towards cancer.